Hey guys, hi, welcome back to the next session of two questions per day here on Unacademy Future Doctors. I'm your educator for biochemistry. I am Dr. Shivangi Shukla. Welcome all of you to this session. Now, I hope this session is more interactive than the last one. In the last session, I had students who were attending the session, but they were not coming up with the answers. Now, the whole purpose of this session, which is designed specially for you to revise, is so that you come up, is so that you interact in the class. Without interaction, what is the purpose of teaching, guys? Then, what is the purpose? If you're choosing to just stay, if you're choosing not to interact in the class, then this one way communication, this one way street is not going to be of any help to you. Okay, sometimes it is better for you to listen to your seniors advice and to take that advice in your stride so that you learn. Okay, got it. Now I hope you guys uh, are more interactive here, and all those out of all those who are attending the session, I am finding them, I should see them answering in the chat section. Okay, if I don't see answers from all those who are present in the session, then I'm really going to take you all, I'm really going to put up a motivational session after this. Okay, or a strategy session. You'll have to tolerate another motivational or strategy session after this, because guys, this is, I'm doing this for your own good. These MCQs, these questions, these tests that I'm subjecting you, they're not going to be of any benefit for me. What am I going to derive? I'm not going to sit for any test. So why am I putting so much energy in spite of being ill? Why am I putting so much energy for you guys? Every day I'm taking 6 sessions, 3 on YouTube, 3 on Unacademy. This is only for you to revise, so that this last week of January is spent revising as much as you can from the course that we have completed. We have completed a very major chunk of the syllabus of biochemistry, that is metabolism. Okay, and in record time. No one finishes this syllabus so fast, and that is why I want you guys to know what are the intricacies in this of this whole metabolism. Now I hope you guys attend this session, and I hope you guys answer. Okay, the whole purpose of this series is for you to be interactive. Otherwise, I'm going to stop this. Otherwise, there's no point in continuing this series, because the whole purpose of an MCQ series is to get answers, is to get students to interact. Anyway, so I'm your educator for biochemistry, Dr. Shivangi Shukla. A bit upset, Shivangi Shukla, right now because of the less the reduced interaction that I'm seeing these days. All right, so all of you would be knowing about the types of NEET PG subscription on Unacademy. If you haven't, you can go and check out the Plus as well as the Iconic subscription. Both will give you access to video lectures from top educators across the country. You get to access a question bank of more than 25,000 questions. You'll get access to live quizzes. You'll get printed notes. And the Iconic subscription will also give you access to resources from Prep Ladder. Okay, so do try these out, and you'll also get an additional 10% discount if you apply the educator code DRSHUKLA. Okay, this code Dr. Shukla, DRSHUKLA, this code is going to fetch you an additional 10% discount. Also, Unacademy has come up with this offer, the Republic Day special offer, where it is going to give you 6 full months free subscription if you take a 1 year or above subscription in the plus category. Okay, so suppose you take a 1 year plus subscription on Unacademy between January 25th and January 27th, that is today, tomorrow and day after tomorrow. Between either of these in either of these on either of these days, if you take a plus subscription of 1 year duration or more, then you're going to get 6 full 3 months extension. That is, a 1 year subscription can become an 18 month subscription. Okay, and that too at the same price. And additional 10% discount also you will get if you apply the code DRSHUKLA, Dr. Shukla. So please try this out, and please hurry up. If you are willing to take subscription, then do it right now. because this is especially important for those who are starting their journey, starting their preparation journey. Okay, so let us start with the question for till this we discussed. Now this is the first question in this session. It is, what is the bond which connects the sugar residue to the nitrogenous base in nucleic acids? Okay, is it A, beta N glycosidic bond, B, acid anhydride bond, C, phosphodiester bond, or D, hydrogen bond? Okay, what is the bond which connects the sugar residue to the nitrogenous base in nucleic acids? Okay, beta N glycosidic bond, acid anhydride bond, phosphodiester bond, or hydrogen bond? Okay, now you will get 5 full minutes to answer this. Take your time, revise, recall the structure of nucleic acids, what are nucleosides, what are nucleotides, what are these, and then we are going to discuss. Okay, do answer in the comment section. All right, your time starts now. 5 full minutes. Come on guys, time is running up. 3 whole minutes are left now. Try and answer, time is running up to answer. The question is a fairly simple one, if I can say, comparatively. What is the bond which connects the sugar residue to the nitrogenous base in nucleic acids? Bond between sugar and nitrogenous base in nucleic acids. Structure of nucleic acids we have discussed in great detail. 1 minute is left now. Come on guys, do not be shy in answering. That is what I always tell you. Do not hesitate in answering. Very good, Jatin has come up with the answer. I'm very impressed with Jatin. He always tries and answers. Very good, Jatin. Very nice that you answer, but let us see if it is the correct answer or not. But I'm very happy that you have attempted the answer in both the sessions. Very good. Let us see if the answer is correct or not. And let us see if there's someone who wants to contest this, who wants to come up with a different answer. What is the bond which connects the sugar residue to the nitrogenous base? Last 10 seconds. Then I'll tell you the answer, okay? Okay, so the correct answer here is A, beta N glycosidic bond. Okay, the bond between the sugar residue and the nitrogenous base is beta N glycosidic bond. Alright, now Jatin, I'll explain why it is not phosphodiester bond and what is the phosphodiester bond. Okay, now first of all, we start with the nitrogenous bases. Okay, for formation of nucleotides, you start with nitrogenous bases. Okay, nitrogenous bases are of two types, purines as well as pyrimidines. Okay, purine is a shorter name. Since it is a shorter name, it will have a larger structure. Larger structure means two rings. A purine molecule has two rings. A pyrimidine molecule, pyrimidine is a longer name, so it will have a shorter structure. Okay, so pyrimidine has a single ring, purine has two rings. Okay, now this nitrogenous base first of all combines with a sugar molecule. This sugar can be either ribose sugar in the case of RNA, ribonucleosides, and in the case of DNA, it is deoxy sugar. Okay, deoxyribose sugar. Now, this sugar is attached. How is it attached? It is attached like this. Can you see? The nitrogen, the ninth atom, this nitrogen here on the, since this is a two-membered ring, this contains a larger structure, two rings. Larger structure means what is this? This orange molecule is a purine, and this green molecule is just one ring. Just one ring means smaller structure. Smaller structure means larger or a longer name is pyrimidine. So this is a purine on the left and pyrimidine on the right. Purine is the ninth, the N9. Can you see? I have mentioned the N9 is connected to the beta carbon of the ribose or the deoxyribose sugar. Similarly, in pyrimidines, the first N, the N1 is connected to the beta carbon of the ribose or deoxyribose sugar. Now, what is the beta carbon here? Beta carbon is this carbon. Why is it called beta? Because the OH group is above the plane of the ring. Had the OH group been below, it would have been alpha. Okay, so beta N glycosidic bond means between beta carbon and N of purine and pyrimidine. Beta N glycosidic. Glycosidic means involving a carbohydrate moiety. Okay, so beta N glycosidic bond is present between sugar and nitrogenous base. How will you remember nucleosides? Nucleoside means they have sugar on the side. Okay, whenever we order coffee or tea somewhere, what do we say? If they ask you, ma'am, will sugar be added or sugar be on the side? So instead of saying please keep sugar separate, you can say sugar on the side, please. Okay, this is a common way of speaking. Sugar on the side. Similarly, sugar on the side in nucleoside. Okay, so this can be a mnemonic for you. Nucleoside means sugar on the side. Nucleoside means sugar plus nitrogenous base. And what is nucleotide? Nucleotide is when phosphate group is tied to it. Okay, and this phosphate group is tied to sugar only. So nitrogenous base tied to sugar, and then phosphate group is also tied to sugar only. Okay, and this phosphate group is tied to sugar with the help of phosphodiester bond. What is an ester? What is ester? Esters are formed by combination of carboxylic acid and alcohol. So the carboxylic acid of phosphoric acid and the alcohol or the hydroxyl group of sugar will combine to form the phosphodiester linkage. Understood this? Okay, Jatin, now your confusion is clear or not? So acid anhydride bonds are between sugar and nitrogenous bases, and beta N glycosidic bonds are between sugar and nitrogenous bases, And acid anhydride and phosphodiester bonds are between phosphoric acid group and excuse me, sugar. Okay. Now, where is this hydrogen bond? This hydrogen bond is present between two nitrogenous bases of the anti-parallel chains. Okay. Okay, this DNA contains a double helix, double helical structure. So the two chains, these two DNA chains are held close together by hydrogen bonds between the nitrogenous bases, complementary nitrogenous bases of the two strands. Okay. Now, A and T will form two hydrogen bonds and C and G will form three hydrogen bonds. And these hydrogen bonds comprise the rungs of the ladder. Okay, the ladder is the DNA and the rungs, that means the steps of the ladder, are formed by the hydrogen bond. Okay, understood this? Now, the next question, which of the following contains desmosines? Which of the following molecules contains desmosines? Is it A. Collagen, B. Elastin, C. Fibronectin, or D. Fibrillin 1? Now, this is a question from extracellular matrix which we have completed long ago in December. So, I hope let us see how many of you remember this. Which of the following contains desmosines? A. Collagen, B. Elastin, C. Fibronectin, or D. Fibrillin 1? Okay. Alright. Now, your time starts now. Try and answer this in the comment section, all of you. Come on, all of you, try and answer this. Very good. Jatin has come up with a very quick answer. Within a minute, Jatin is quick with the answer. 4 minutes are left now. The others can also try and attempt this. Come on, try and answer this, guys. Last 2.5 minutes left now. Okay, time is almost up now. 30 seconds and then I'll tell you the answer. Whether Jatin is right, let us see. Okay, Jatin, I'll be coming up with videos on xenobiotics and cancer drugs. That is actually a very small part of the whole biochemistry syllabus. So I wanted first to get your concepts cleared on metabolism, nucleic acids, all these topics, hormones, clinical biochemistry. I'll come up with questions on with the discussion on xenobiotics and cancer also. Okay. Okay, I've taken this up as your feedback, and I'll take classes on that as well. Okay, do stay tuned. There are a lot of quizzes coming up, so do stay tuned to all of this. You'll be very positively motivated. And any question that you get wrong, anywhere, whenever you're revising, if there is something that you cannot recall, just go back and watch that session at 2x speed. Okay, or go back and watch read your notes quickly, skim through them, then so that then and there you solve all your doubts. Then and there every difficulty is solved. Okay, otherwise if you keep it postponed, if you procrastinate, then you are never going to go back and read that up. Okay, so please try, because all these short topics, no, like immunology, xenobiotics, cancer, digestion, all these small topics, they'll be very easy. Okay, also homeostasis, we have discussed a lot of questions on homeostasis. So if you guys want, if you want some more discussion on homeostasis, then I'll take that up also for revision, because homeostasis is again a very important topic. It is all understanding based topic. Okay, it is very, very important. All right, so now I'll tell you the answer. It is very good. Jatin is very correct. Congratulations, Jatin. Congratulate yourself. Let us clap for Jatin. The correct answer is elastin. Okay. Desmosines are unique to elastin. Now, elastin is what? It is a structural protein present in extracellular matrix. Now, what is the extracellular matrix? Extracellular matrix is the connective tissue present outside the cells, in between the cells, that binds the cells together and prevents them from moving about. Okay. Now, this extracellular matrix also helps in transport of substances from one cell to the other. Okay. Now, this extracellular matrix contains one, structural proteins. These structural proteins are of three types: collagen, elastin, and fibrillin 1. Okay. Next, it contains specialized proteins. These specialized proteins are of two types: fibronectin and laminin. And the third type of biomolecule that it contains are proteoglycans. So extracellular matrix contains collagen, elastin, fibrillin 1, fibronectin, laminin, and proteoglycans. Okay. So elastin is one of the structural proteins present in the extracellular matrix. It is present wherever elasticity or extensibility is required. Okay. Collagen gave strength and rigidity, strength to wherever it was present, strength to the tissues wherever it was present, while elastin gives extensibility, or the property of extension, elasticity and coming back. Okay, getting extended and then coming back. This elasticity is provided by elastin, and it is present wherever you need some elastic component, elastic properties, that is in the lungs, in the large arteries, in the ear cartilage. The ear cartilage of the external ear is made up of elastin, and also it is present in the skin, because our skin is what? Elastic. It goes back, isn't it? So our skin is elastic, and as we age, this elastin and collagen, it tends to break down, and its synthesis tends to slow down. Therefore, the elasticity and the strength of the skin and the suppleness of the skin reduces. Okay. So, Acha, tell me, what is the cartilage present on the tip of the nose? What is this cartilage? The cartilage in the ear is elastin. What is the cartilage at the tip of the nose? Can you tell me? Jatin, can you tell me what is the cartilage at the tip of the nose? Okay, the cartilage at the tip of the nose is the hyaline cartilage. Okay, understood? That cartilage is hyaline cartilage. You would have read about this in anatomy. In the first year, they teach you about the different types of cartilages present at different locations. Alright, now this elastin is synthesized as tropoelastin. Jatin is asking what are his rewards. Jatin, keep on answering more and then cumulatively I'll give you guys rewards, okay? But you have to answer more. This is just the first session. No, you attend all the sessions and answer in all the sessions. Okay, and then we'll put up a quiz, we'll rank winners and then we'll reward them also. Okay? Now, elastin is synthesized as tropoelastin. Tropoelastin is what? It is a soluble form of elastin, okay? And this has to be made insoluble for it to be present in the extracellular matrix, okay? Now, this tropoelastin, how is it different from collagen? What were the different important components of collagen? They were proline, glycine, hydroxyproline, lysine, hydroxylysine, and glycosylated hydroxylysine. You remember how hydroxylysine residues were glycosylated also there. So therefore, it was a type of a glycoprotein. Why? Because it had carbohydrate moieties also. Whereas in elastin, what is there is proline and hydroxyprolines are there, alright. But lysine is there, okay. Proline, hydroxyproline and lysine, all three of them are there, but hydroxylysine and glycosylated hydroxylysine are not there. So elastin does not contain carbohydrate moieties, any glycosylated moieties. Therefore, elastin is not a glycoprotein. Understood? Very important difference. Next, in collagen, you had in collagen, what did you have? High important points. Hello, please concentrate on this. Okay, we are discussing the structure of elastin. Do concentrate on this. Now, the lysyl residues or the lysine residues present in collagen, they underwent oxidative deamination reaction. Oxidative deamination reaction to form aldehydes. Okay. Now, these aldehydes are modified lysine residues. Okay. Now, these modified lysine residues, that are these aldehyde molecules, they were combining together between the chains to give strength to the collagen fibril. Okay, the modified lysine residues were combining together to give strength to the collagen fibril. Now, what happens in elastin is the same enzyme, lysyl oxidase. This will oxidatively deaminate lysyl residues in elastin also. It will form aldehydes in elastin also, just like collagen. But the bond, the interlinkage between the chain, the cross links are between one unmodified lysine and one modified lysine, one aldehyde form of lysine. Okay, understood? So in collagen, both were modified lysine residues, but here one unmodified and one modified residues are connecting together. This kind of a linkage is unique to elastin between one unmodified lysine and one modified lysine. This kind of a cross link is called desmosine. Okay. Okay, this kind of a linkage is called desmosine, and these desmosines are what will lead to the elastin becoming insoluble. Earlier, what was elastin? Elastin was tropoelastin. Tropoelastin is soluble. Now, this desmosine, that is the linkage between one modified lysine and one unmodified lysine. This desmosine is going to form an insoluble elastin molecule. Okay, this insoluble elastin molecule leads to stability of the elastin. Okay, understood this? Got it? So this was about the structure of elastin, how different. Okay, one more very important thing is that this lysyl oxidase enzyme has the cofactor, that is, it requires copper as the cofactor. Okay, so whenever copper is absent, like in Menkes disease, whenever copper is absent, then this lysyl oxidase cannot function properly, and what will happen? Lysyl residues cannot be converted to the modified aldehyde forms, and these desmosines are not formed. So the tropoelastin cannot convert to elastin, and what happens is the connective tissue is weak. Similarly, the collagen is also defective because the cross links, the cross chains cannot form because of deficiency of copper and because of decreased functioning of lysyl oxidase, and therefore collagen is also weak. Okay, so deficiency of copper in Menkes disease and other nutritional deficiencies is going to lead to defective connective tissue. Okay, understood? Yeah, very good, Jatin. All right, then. Thank you, guys. Thank you for this session. Now we'll meet in another session in a few minutes, and that will be the third such session. So we'll discuss the last two questions for today. Okay, in that session. Okay, guys. Bye bye. Take a break, have some water, and then we'll join in the next session. That is on 2 questions per day. That session. Okay. All right. Bye bye, guys.